はい、津本でございます。本日の、この勉強会の座長を務めます津本です。よろしくお願いします。それでは、本日のセミナーについて、ですけども、まずこれ英語講演になりますのでですけども、最初に今日の演者でいらっしゃいますウルリヒ・ラント博士はですね、日本語で、こうご紹介したいと思います。よろしくお願いします。ラント博士はですね、2005年に、ミュンヘン工科大学で、生物物理学において博士号を取得されて、その後ミュンヘン工科大学で博士研究員を務められてます。で、一時は富士通研究所においても、研究をされたこともあるというふうに伺っております。2007年に同大学でグループリーダーとなられて、ドイツにおけるエクセレンスイニシアティブですとか、ミュンヘンクラスターにおけるナノシステムイニシアティブにおいて、首席研究員として活躍されました。その後、ドイツの科学省からバイオテクベンチャー創生を支援するグラントを獲得されて、2012年に生体分子間相互作用解析であるスイッチセンス技術に基づいて、ダイナミックバイオセンサーズ社を創業されました。これまでに CEO としてこのナノ分析装置ですとか、バイオチップの継続的な開発やマーケティング、それから欧州、北米、それからアジアにおけるビジネス展開をリードしてきておられます。また、研究者としても分子間相互作用ですとか、ナノテクノロジー分野で70本以上の強調論文を発表してきておられます。本日はご自身が開発されてきたスイッチセンス技術を応用した細胞表面タンパク質の分子間相互作用解析を中心に、免疫療法や細胞療法の開発においても、活用可能なバイオチップの例など、多くのアプリケーションについても詳しくお話をいただく予定になっています。はい、それでは始めたいと思います。最初、少しドイツ語で言ってみますが、失敗するかもしれません。はい、では、ドクターランテDr. Rant, can you hear me?
Yes,
Vielen Dank
yes,
für
sorry.
Ihren heutigen Vortrag, right? 聞こえるかな?
Yes.
Yes. So, könnten Sie bitte mit Ihrem Seminar beginnen?
Oh, vielen Dank für die introduction.
Yes.
Thank you very much, Tsumoto-sensei, for the very kind introduction, and thank you also, Yumoto-san, for inviting me. It's a great pleasure to have been given the opportunity to present in front of you and share some of our latest results that we obtained at Dynamic Biosensors. So I'll share my screen with you. I hope it works. I hope you can see my presenter screen now. Yes.
Yes, yeah.
Wonderful. Okay, so the title of this presentation is quite long. I would like to present something to you about the next generation instruments and biochips to study and investigate molecular interactions. And we do that, as I said, on chips and recently also on cells, especially cancer cells. And our goal is to advance immuno and cell therapies in that manner. So the contents of today's talk is like this. I would like to address these 3 points. I'd like to give you a short intro into why we think real-time biosensing is actually relevant in the drug discovery process and what are current challenges in that respect. And then I would like to introduce 2 different technologies to you. The first technology is called Switch Sense, and it enables one to measure interactions, binding parameters between 2 types of molecules. So a molecule-molecule interaction analysis tool. And the cell trap technology, as you can already guess from the name, is intended to measure interactions on cells. So we can then follow in real time how molecules bind or unbind from cell surfaces. Before I get started, I just would like to give you a little bit of background on who Dynamic Biosensors is. So we are a team of more than 60 life science specialists. We are located in Munich, in Germany, right in the center heart of Europe, if you will, and have recently also opened an office near Boston in Massachusetts in the USA. Our field is obviously life science tools. I will show you that we make instruments and develop new technologies to measure interactions of molecules. We cater to the pharmaceutical and to the biotech industry, as well as to academic customers, and are very proud to have renowned research institutions like the Tsumoto Lab at the University of Tokyo, but also others in our customer base. The two core technologies that we are investigating I have already mentioned, and I will show you later how this is being done in practice. So if you think about molecular interactions from a very bird's eye view, you could say, so what is interesting with respect to molecular interactions between proteins, small molecules, nucleic acids? So first of all, you could think about binding. If you think about binding, the first thing that comes to mind, what is the binding strength, the affinity or the avidity of an interaction? And then when it comes to real-time binding, what are the kinetics of an interaction? How quickly does a reaction occur, and how stable is this reaction? There's also the element of structure and function when you think about molecular interactions. So very often conformational changes occur in proteins or nucleic acids, RNAs, for instance, when they bind a small molecule or another protein. And then, of course, there's also the element of multimerization, that protein complexes are formed or things like that. And there's many more things to think about. So if you look at the issue in a little bit more detail, and you consider one interaction, here's an example of a DNA binding protein that binds here to a DNA strand. So what is the first kind of information you would like to get? That's probably a yes or no answer. Is it binding or not? And that is typically done during a hit screening assay in a drug discovery process. Then when you try to gain more information about this interaction, you would be interested in the concentration dependence of this binding. That would then result in the analysis of equilibrium binding constants like the KD value or IC50 values. And then the next more complicated step is to observe this binding in real time and also measure and gauge the stability of this interaction. And that is then what we would call association and dissociation kinetics. Now, things get more complicated and more complex to be measured as we move up along this ladder here. Now, if the interactants actually have more than one binding site that engages in the binding process, then one refers not to affinity, but to avidity. And there could be ternary complexes that are formed, or even higher order complexes, interlinking occurs, and so on. I mentioned before already, very often binding has an effect on structure and that again affects the function. So conformational changes might occur in these proteins or nucleic acids, and that might affect the enzymatic activity. And then one of the holy grails over the last couple of years really is to measure directly on cells, not just equilibrium binding constants, but also to attempt to observe the binding in real time and again extract association and dissociation kinetics. So I'd like to actually now talk a little bit about this central point here, real-time binding and stability measurements. And I'd like to briefly introduce how this is being done with a biosensor. So very simply put, many surface biosensors work in that way. You have a flow channel, you have some ligands here in pink that are immobilized on the sensor surface, and then the instrument, the apparatus, exposes these ligands to analytes. It flows analyte molecules here in blue across the sensor surface, and then they are allowed to bind to the ligands on the surface. And after they are being washed off, they are dissociating from the surface again. And in terms of the real-time binding signal that one attempts to observe, that very often looks like this. So you try to measure something like a fraction bound that could be a surface plasma resonance signal very often, or in our case, as you will see later, that is a fluorescence signal, but it is always related to the fraction bound of molecules on the surface. And then when you inject the molecules, you observe an increase in the fraction bound. And then when you wash off the molecules, you observe the dissociation behavior. And then you use mathematical models with, in the ideal case, very simple equations, just exponential functions, to model the decay of the fraction bound or the association phase. And let me give you a very concrete example now how this looks from an experiment that is conducted with our switch sense sensor. So this is like a model interaction, a short DNA strand, two DNA strands that are allowed to interact and form a DNA duplex. In this case, it was a 9 base pair DNA. You can see how these experiments are typically conducted. It's kind of good practice to inject not one concentration, but several concentrations of increasing magnitude into the flow channel and repeat the experiment, and then run a so-called global fit analysis where all these dissociations are fitted at the same time, and then all of these associations are also fitted at the same time with these exponential functions, and the concentration enters as a known parameter here. And what we obtain from these experiments then is the association rate, so how quickly do these molecules bind to each other, and the dissociation rate constant, so what is the stability of the interaction, how quickly do they fall apart. And the dissociation constant, the equilibrium affinity, if you will, is actually related to these rate constants. So if you know the rate constants, you can also calculate the KD value or the affinity of this interaction. Now, why is it important not just to know the KD value, but actually to know these association and dissociation rates? And I picked an example from a very recent drug discovery project that we were involved in with one of our customers. I think it's a nice example that shows that you can measure similar KD values for interactants, but have very, very different kinetics. So you can see here two different small molecules, or I call them red and blue compound here. So these are small molecules that inhibit an enzyme. In this case, it's a protein ligase. And these small molecules were measured with our switch sense sensor. You can see here a fluorescence change. So the signal is decreasing here due to a fluorescence quenching mechanism. I will explain that in detail a little bit later. And then we can extract from these association and dissociation rates. We can plot them here in what we call a rate scale plot. So we have the association rates, the on rates up here. We have the dissociation rates, the off rates down here. And then we simply draw 2 points and we connect them by a line. And the length of these vertical line here tells you what is the affinity of the interaction, what is the KD value. It's also written here. You can see both of these interactions have actually a KD value very close to 80 nanomolar, but you can see also that the on rates are very different and the off rates are very different. So both of them differ by approximately a factor 3 to 4. And in fact, the red compound with the slower dissociation, so the higher stability that binds tighter, is actually the one that also showed a superior efficacy in a vivo system in a cellular assay. So in this case, the KD value would not have been predictive to select the red compound, but the kinetic analysis is. Now, where are these kinetic measurements and other biophysical characterizations being used in the drug discovery process? You can see here a simplified diagram of how such a drug discovery process works, from the target identification and the hit generation phase that might involve thousands or even millions of compounds that are kind of screened against a target. Then before the clinical phases and the in vivo studies are being performed, a biophysical characterization is usually performed. And during that biophysical characterization, a lot of information, useful binding information and information on potentially conformational changes, temperature, behavior, and so on is being analyzed so that the drug candidates that are moved forward and advanced in these pipelines are well understood, and that a high success rate is actually being achieved in later very expensive phases. And the average costs for a new chemical entity or new biological entity to reach the market now has exceeded $2 billion nowadays. So it's a very expensive business. And the message is really, if we can analyze these molecules very well in early stages, then we will have a faster and more successful drug discovery process. Now, the way we do that is with these products, and I apologize for the product placement here. It's the last slide I will show you about what Dynamic Biosensors actually makes, but I think it's important to appreciate we make these instruments that are electro-optical analyzers. I will explain in a couple of minutes how they work. They also need biochips. I will also explain how the biochips are being functionalized, and then I will not discuss the sample preparation too much in this talk. But this is also something that we have solutions for. Now, I mentioned that we have these two different technologies, switch sense and cell trap. And one, the switch sense that I will address first, deals with molecule-molecule interaction. So here is an example of an enzyme, a polymerase that binds to a DNA strand. So we do a lot of work with DNA, RNA binders, but also with protein-protein interaction, small molecules. And in this talk, I will mostly talk about, speak about multi-specific antibodies and small molecules. And in the second part of the talk, I will then speak about cell molecule interactions. And that is relevant again for immunotherapies, CAR T cells, T cell engineering, cytokine release studies, all things that are related to the analysis of cellular behavior. Now, why do we think it's actually necessary to develop new enabling technologies for drug discoveries? I picked a recent review article in Nature to exemplify this, and Mr. Deshays from Amgen explains here the different waves or phases of drug discovery over the last decades. And he describes that in the 1970s, we have seen the rise of rational drug discovery. And then in the 1980s, here we've seen the invention of protein-based therapeutics, and then especially antibodies coming to the market. And so the future, what can be observed already is that these, especially protein-based therapeutics, but also small molecules, are becoming smarter and smarter. And by smarter, I mean they are targeting not just one target, one protein in the body, one DNA strand, but they are actually multi-specifics and have very interesting and complex mode of actions. And here are shown two examples for a small molecule. You could call them a matchmaker. So that would be an example where a small molecule brings an effector protein to a target protein. I will give you an example later from the field of PROTACs, where this is a ligase that then modifies the target so that the target would be actually degraded by the cell itself. So this is an example of, I would say, a sophisticated small molecule. Here are examples for antibodies that would connect T cells from a patient's immune system to a cancer cell. And obviously, what is really important here is that the T cell is then connected not to a normal cell, to a tissue cell, but recognizes the tumor cell with very high specificity. So what is needed is not just a connector, an interlinker of these two cells, but what is needed is an interlinking molecule that is very specific to the cancer cell. And very often nowadays, researchers are looking into ways of engaging not one, but two or more targets on a cancer cell in order to be very specific about the recognition of the cancer cell. So these are interesting molecules, and I would just like to mention that these are also molecules that are making a lot of money for pharmaceutical companies. So the global antibody market has already exceeded the $100 billion annually. But what's also interesting is, if you look at this graph here, the number of bispecific antibodies that are in the clinic. We are currently seeing more than 100 of these bispecific and multispecific formats that are being tested in the clinic, and hopefully will reach applications and become therapeutic soon. We also see that the publication numbers on bispecifics are increasing quite dramatically over the last couple of years. So how do we come into play here? I will now show you how SwitchSense works, and these are probably the three, I would say, prime examples where SwitchSense is being used. I will only have time to address one of those today very much, but I wanted to mention that we work a lot with DNA and RNA binders and enzymes. This is something that we're very good at. We also do studies of conformational changes in proteins. You can see here an example of a protein that's related to the innate immune response called STING, and you can see the overlay of two conformations, and SwitchSense can be used to detect not just the binding of a small molecule to this protein, but also the conformational change that is induced by the binding. I will only be able to touch briefly on this. I will mainly talk about multi-specific molecules like bispecific antibodies that can engage several targets at the same time. Now, how does SwitchSense work? I would like to start off by showing you how the surface is being structured and what the architecture of molecule immobilization is on our surfaces. So this is our instruments. Down here is where the samples go. That could be a 96 or 384 well plate. And up here we have a rack of 5 chips that the instrument can exchange automatically. This is how such a chip looks like. What's important is really the central part here. So you can see beyond these metal chip cover, you can see here a glass microchip with a microfluidic channel in the center. And if you would zoom in into this central hole here, you would see 2 spots that are about 100 micrometers in diameter. And on these 2 spots, we actually record 4 signals. I will explain later how these 4 signals are being recorded, but for the time being, I would like to draw your attention to how we immobilize molecules on these spots. So what we put on these surfaces when we produce these chips is a DNA molecule that we call the anchor strand. It's typically a 48 base mixed sequence of DNA. And then onto this anchor strand, the instrument, the user, can hybridize and bind different other strands. So we usually use one strand that we call the adapter strand that connects the strand with the ligand. So that ligand here, our penta, our pink molecule here would be the one molecule of interest, and the analyte in blue would be the other molecule of interest. That could be, as I said before, it could be a protein, a peptide, a small molecule, or an RNA DNA sequence. And one of these molecules needs to be attached to our surface. And that can be done by a DNA strand that we call the ligand strand that is then bound to the adapter strand and connected to the surface. You can also see that the adapter strand has a fluorescent dye, and this dye is being used for detection. I will explain that in a minute. Now, how do molecules get to the surface and get away from the surface again? We have a very simple way of regenerating the surface. As you've seen before, the surface itself is just functionalized with the anchor strand. Then the ligand is being hybridized onto the surface. This is an automated step that just takes a couple of minutes. Then the actual experiment is being performed. So the blue molecule is being injected in the flow channel and is allowed to interact. So we measure the association, and then we can also measure the dissociation. And then in the end, we can, if we would like to remove and refresh molecules from the surface and refresh the surface, we can then wash off these DNA strands simply by a sodium hydroxide wash, and that denatures the DNA and gets off everything. The way that these molecules are bound, the ligands, so a protein, for instance, is bound to this ligand strand here, to this DNA strand, is either through a covalent coupling step, so that might be an amine that is used here, or a thiol group, or we use capture methods like his tags, biotins, and others, strep tags, very, very standard coupling and capture strategies. Now, why do we use these DNA strands? You can see here a nice example of how you can play with the surface density of these ligands on the surface. And that is, in many experiments, that is very important, because you would like to control the distance between these molecules so that you can allow them to be interlinked or not be interlinked. And the way this is being done in our experiment is very simple. The strands are just being mixed at a desired ratio, let's say at 1:1, then you will create a surface like that. Or if you would mix them at 1:50, you would then also create a surface where only 1 out of 50 DNA strands on the surface actually carries a ligand. And so the dynamic range of these surface density variation is actually above 200. So we can vary the density by a factor of 200 very conveniently, simply by mixing these strands together. And that is actually being guided through an app that one can download on our webpage. Now, you can not only mix a strand like shown here, a ligand, a strand with a ligand, with what we call a dummy strand here that does nothing on the surface, just to dilute it, but you could also mix 2 different ligands. And this is where our fluorescence approach comes into play. I mentioned before that we have a dye at the end of our adapter strands. Now, this dye can either be a red or a green dye. Actually, we have different green and different red dyes in our portfolio. But the important message is we record green and red fluorescence at the same time. And so now you can put 2 different species of molecules on the same surface, and you can record and discriminate binding signals from species A versus species B. So it's not like a refractive index sensor like an SPR, which will record a binding signal as an average over the whole surface, but it will be able to discriminate whether it is ligand A or ligand B. And in fact, I will show you later that this way of immobilizing molecules is a very flexible means of doing so. So I will show you later other simple DNA nanostructures that can put on the surface. I will talk a lot about this DNA Y structure today, but you can also put DNA origami nanolevers on the surface if you would like to study very large analytes. So we have customers who even study ribosomes at 2 megadaltons with this approach. Now, let me explain a little bit about how the measurement actually works. You have already seen that the molecules are coupled to the surface via these DNA strands. We call them DNA nanolevers. And you will see in a second why we call them nanolevers. So in fact, we have different measurement modes. We have a dynamic measurement mode. We have a static measurement mode. And then we can do that in 2 colors at the same time as we've discussed before. Let me start off by explaining the dynamic measurement mode, because this is where the name switch sense actually derives from. So what we do here is we apply alternating voltages to the microelectrodes, and that actuates these negatively charged DNA strands on the surface so that they would start switching their orientation up and down. And at the same time, we will measure these orientation switching by recording the fluorescent light emitted from this dye at the end here. And let me show you this simple cartoon that illustrates how that goes. So the surface here is charged positively and negatively. When it's charged negatively, the DNA is repelled from the surface. When it's charged positively, it's attracted. And you can see that the fluorescent light from this dye up here is actually being quenched by the gold surface. So we're taking advantage of a fluorescence resonance energy transfer. It's not a threat between 2 molecules, but it is between the molecule and surface plasmons in the gold surface. So it's very long ranged. It works from 0 up to 200 nanometers into solution. So we can measure the orientation very accurately. And what you can also see from these examples here really is that if you immobilize then something small onto the surface, the switching speed will still be relatively high. Whereas when you have a large analyte, then the switching speed will be slow. And that is simply not a moment of inertia effect, but it's hydrodynamic friction. So we're moving these DNA molecules with their bulky heads, with their wobble heads through the solution, and the hydrodynamic friction in the solution retards the motion of the molecules. Now, the way this is being done in terms of measurement technology is briefly described here. So we have these different states, the quenched state, and then a gradual increase in fluorescence as the DNA nanolabel moves to a more upright position. And the way it's being measured is we continuously excite these fluorophore up here, and then we measure the photons that are being emitted from these dyes with single photon counters. And now the twist of the measurement technology is that an electrical trigger that comes from the alternating voltage here will then be correlated with the photon arrival time. And that means that we know when the DNA is repelled from the surface and how quickly it is being repelled from the surface. So on a sub-microsecond timescale, we can follow the motion of the DNA as it stands up on the surface, then is in the upright position, and then as it comes down again when a positive voltage is being applied. So in this process, the instrument actually acquires a dynamic information, a switching speed, and it acquires steady state fluorescence values in the lower position and in the upper position. Now, this is being used actually to measure also conformational changes in molecules. I will not have the time today to go into this in much detail, but I would like to explain the other measurement mode instead. So the other measurement mode that is actually very, very simple is what we call the static mode. So there is no voltage being applied, small negative voltage, usually just to repel the DNA from the surface so that it stands on the gold, and is kind of good, very well accessible to binding from molecules that come from solution. Now, what we use here is we just look at the fluorescence, the absolute fluorescence of the dye molecule that's attached to this DNA. And these lines here should indicate that we actually have collisional quenching between the dye and anything that is close to the dye and is close in the chemical environment of the dye, and that will always give rise to changes in the fluorescence. Usually we see a quenching of the dye fluorescence, but sometimes that can also result in a enhancement of the dye fluorescence by typically a couple of percent up to 10, 20, 30, 50%. So changes in the dye fluorescence will tell us whether something has bound in its vicinity, in its proximity, and this is why we also call these fluorescence proximity sensing. And I would like to give you now an example how this works. Very simple example, a DNA-DNA interaction assay setup. So here is an example where not a protein has been tested, but just the binding of short DNA strands to an overhang here have been tested. And the idea behind this experiment was to test how low can we go in binding affinity so that we can still measure an interaction here. So we started off with a 9 nucleotide target and then went down to 2 nucleotides. And so we cut these DNA target by 1 base at a time and measured the kinetics. And then you can actually get sensorgrams that look like this. So if you start off with 9 base pair interaction, you can see here the associations and then a slow dissociation. And when you reduce the number of base pairs to 8, 7, 6, you can see how the dissociation becomes faster and faster because this interaction is getting weaker and weaker. And this is also why we chose DNA here as a model system, because you can appreciate that fewer base pairs confer a lower binding energy. And you can see here that the very quickly at around 6-5 base pairs, the sensorgrams almost look rectangular. And there's very fast kinetics in these edges here. And then at 3 base pairs, we hardly can measure anything anymore. And at 2 base pairs, even up to a concentration of 3 millimolar, we couldn't measure an interaction anymore. Now, if you look into these edges, now here's the 9 base pair to 7. These are really slow dissociations on the time scale of hundreds of seconds. But if you look into the shorter DNA, you can see that when you sample with 100 hertz, so 100 data points per second, you can still resolve the dissociation of very weak binders. And now if you plot this dissociation rate constant, you can see that the dissociation rate becomes faster and faster as the number of base pairs is decreasing. And the state of the art of other biosensors, the limit that they can access, for instance, Biocore mentions that the SPR systems can resolve a dissociation rate up to 2 per second. So that would be around here. And you can see that our fluorescence sensor with these high performance fluidics can record even rates up to exceeding 20 or 30 per second. So that's pretty good. I'd like to show you that this also works for not for DNA, but also for, here's an example from Roche, where they studied the neonatal FC receptor binding to antibodies. So they were interested in actually the FcRn receptor facilitates the recycling of IgGs in the cell by rescuing them from lysosomal degradation. So that's an important property when you think about the residence time, the survival time of an antibody in the body. And they engineered their antibodies so that they would use that rescue system very efficiently. And you can see here that sensograms have been recorded at different pH values. At a low pH, you can see that the residence time, the binding is actually pretty strong, whereas at a high pH, the binding is very fast. So we're talking about hundreds of seconds here and tens of seconds there. And again, if you use our rate scale plots, so here's again the on rates and the off rates, and you draw the affinity vertical lines here, you can see that the on rate actually almost stays constant over a change in pH value, but the off rate becomes faster as the pH value reaches 7. So the stability of this interaction is compromised by the pH value. And it's not so much the propensity of this interaction to occur, so the association rate stays the same. Now, I would like to go beyond one-to-one interactions and talk a little bit about bispecific binders. And there's some very interesting examples now in recent therapeutics coming up. One that has been developed in Japan by Chugai, and I will show you in the next couple of slides some measurements that we did with the bispecific antibody emicizumab or Hemlibra that treats a bleeding disorder by substituting a missing cofactor. Yeah, so very good in that respect. But there's also other examples, especially in immunotherapy, lung cancer, for instance, where bispecific antibodies are engineered to bind to 2 targets on the same cell surface. And if you just look at the problem from a very mechanical point of view, you have these 3 states or 4 states. You have an unbound state of these targets on the cell surface. And then when the antibody comes in, it could either bind via antigen A or bind via antigen B, the red or the green. And then it will form a ternary complex, or it can form a ternary complex. In the case of the Hemlibra antibody, you will see this is actually the biologically active state. So this is the state, the ternary complex that needs to be achieved. And now it can, of course, because we're in a dynamic equilibrium, it can go back and forth between all these states. So it can let go of one arm and then re-engage to a ternary complex, or the other arm and re-engage into a ternary complex. So these are actually dynamic transitions that are taking place here. And from a measurement perspective, it's more complex because now we have to discriminate the binding to the red and to the green antigen. And the way that we are doing this, again, now you already know how the sensor surface is being functionalized. So there's our anchor strands on the surface, and now we use what we call a Y structure. very simple DNA structure that is just a fork of a DNA strand. And then we can functionalize and immobilize one antigen to one arm and the other antigen to the other arm. In this case, for Hemlibra, that was factor 9 and factor 10 that were immobilized on these two different arms. And there is a flexible hinge region, so these arms can actually move around and they can present the antigens to the antibody in an accessible format. So it can always bind to this antibody. And now I'd like to share some of the results here with you. You can see that we repeated these experiments very quickly and easily in different combinations. So one binding situation would be that only one factor is being immobilized. The other situation would be the other factor is being immobilized, and then we can put both factors on the surface. And this is the binding from our green antigen, factor 9. This is binding to factor 10. And then this is the signal that we get when both factors are on the surface. And you can see quite interestingly that factor 10 is not really affected much by the presence of factor 9. So these two binding signatures look quite similar. Whereas for factor 9, you can see a very distinct difference. So especially at lower concentrations, like here, you can see that a slow dissociation occurs that is not present here. At high concentrations, you can see that a fast component is still present or is again being present. And now I can't go through all the details here now, but again, we can plot the affinities, the on rates and the off rates here from the left side, from the right side, and when both of them are present at the same time. And you can see that factor 10 in orange here doesn't care too much about the presence, whereas factor 9 in green and blue here actually develops a binding mode that is very similar to factor 10. So what we can conclude from that is, first of all, this antibody recognizes its targets with a very markedly different affinity. So it binds to factor 10 100 times stronger than to factor 9. And it is probably happening that it at first engages factor 10. It binds with the orange arm first, and then is being handed over to the blue arm and then binds to factor 9. Now, this is quite interesting because it is related to the biological activity of this antibody. The catalytic reaction that is actually occurring by factor 9 has a half lifetime, if you will, of about 15 seconds. And factor 10 holds it in close proximity to factor 9 for about 20 seconds. So this is kind of the switch that allows factor 9 to perform its enzymatic activity. And that's kind of the secret of success of why Hemlibra is working in patients. Now, there's also a quite interesting finding. If you look at the concentration dependence of these curves here, the higher the concentrations get, the more pronounced this fast decay here, the fast dissociation happens again. And that indicates that we actually not only have the ternary binding mode anymore, but we have kind of an overloading of the system. And we have these 2 times binary binding at very high concentrations, which for the clinician is a very interesting red flag. It tells us you should not overdose this antibody in a patient because it will not just not be better, it will actually reverse its action and will become less efficacious. So this is a quite interesting finding that we can derive from the binding kinetics here. Now, I'd like to give you an example of a small molecule that has bispecific components. This is a so-called PROTAC, a protein degrader. So what happens here is that a protein of interest is being connected to a ligase. In our case here, a VHL or VCB, and also cereblon. I will show some data about this. And then these ligase ubiquitinates the protein of interest, thereby labeling it and tagging it for destruction by the proteasome complex in the cell. So this is a new way of how drugs can work in the body, that they would actually label other proteins for degradation. And the question here is, how can you measure such a ternary complex? Because now you have to connect a ligase and a protein of interest through a small molecule called a PROTAC. And again, we're using our Y structure here. And here you can see it's a dynamic equilibrium. The Y structure can open or it can close. And what's really a nice measurement mode here is now, again, in the different situations, we only functionalize the left arm with one protein or the right arm with the other protein. And in the middle, you can see the situation where both arms are being functionalized with the protein. And when we come in with the small molecule, you can see now in the red and in the green fluorescence that here, of course, we only get green fluorescence. Here we only get a signal in the red fluorescence. And here we get a counteracting signal in green and red. And the reason for that is that we're actually using now a fluorescence resonance energy transfer, a FRET. We excite the green fluorophore, and then the energy is transferred to the red fluorophore, and we can read that the presence that these two fluorophores have come into close proximity, and that is due to the connection of these two proteins through the PROTAC. So that is a very nice way of showing in a functional assay that it's not just a binding event, but that this binding event actually moves these two proteins together, and that it's not just a binary complex that is being formed, but we can confirm that this is a ternary complex. And again, we can compare then the KD values of the two binary components, so a 12 nanomolar and a 140 nanomolar, when you can see here now the avidity component is actually stronger than both of them, which was expected for this case. Now, why is that interesting? Not just to study one PROTAC, but you can now do that, of course, in a well-played format. I mentioned we can record in real time 4 signals, 2 colors on 2 spots. And you can see now when you screen a 96 well-played with different compounds, you can immediately, just by a bare eye, you can kind of identify the binders. So here, nothing happens in the blanks. This is a FRET signal. You have these counteracting red and green signals. And here is a signal for MZ1 that only binds to the green arm, but doesn't bind to the red arm, and so on. And from this kind of high throughput data, we can then measure or extract very useful information. So we can plot these as a map, which compounds generated a binary signal and which compounds generated a ternary signal. You can see here that there's a heat map for some compounds that actually have a ternary component. Others have, these ones are negatives. These ones are components, compounds that only bind in a binary mode. And then we can even run a p-value analysis of this so that we can gain statistical, or we can identify our hits with statistical confidence. And you can see here that these here are the hits that have a high statistical confidence, a very low p-value from the no hits. And then for these, we can also extract already kinetic rates, and we can plot then the off rates versus the on rates. And we can learn a lot about the stable binders, the fast engagers, the slow engagers, and we can actually rank these binders in a drug discovery approach. So you can extend these interactions to high throughput. Now, in the last couple of minutes, I would like to address a new technology. Everything that we've seen so far has been shown on biochips. Now I would like to move on to cells. And why is that interesting? Obviously, if you take a molecule and you measure it on a biosensor, it is not in its native environment anymore. If you think about membrane proteins on cells, they would like to be in their membranes and be around the other molecules, the other cofactors maybe that are present on the cell surface. So the measurement of kinetics on cells is something that has been attempted for quite a while now. And the goal here is to measure, again, the association rate and the dissociation rate, but now directly on cells. And this would have implications for immunology, drug development, for adoptive cell therapies, if you think about CAR Ts or engineered T cell receptors, but also for cell-cell communication, for instance. And again, why is it so relevant? I'm not going to go through this busy slide. Just if you look at these examples here that I've already discussed before. So if you think about connecting two cells, then this connection, this linking will depend on the binding kinetics of these individual binding moieties of these T-cell engagers here. Very, very crucial. Yeah. So these need to be characterized in order to have a designed and well-defined engagement of a tumor cell by a T-cell. Now, you can not only think about conventional antibody interactions, but you can think about, in a very general sense, I already mentioned chimeric antigen receptors or a TCR MHC interaction. So there's actually a quite vast literature out there that tells us that it's not just about, if you think about, for instance, MHCs and T-cell receptors, how they interact, how T-cells recognize foreign antigens. The kinetics of these interactions are actually very, very crucial for a highly efficacious T-cell approach to a cancer cell. So knowing these kinetics is really important. Now, why is this difficult to get? We have already mentioned the surface plasmon and resonance sensors in the past. So this is obviously a technology that has been around now since the 80s to measure real-time kinetics. But I will show on the next slide, it is actually difficult to immobilize cells on SPRs. The other technology that's super important in cell analysis is obviously flow cytometry. And it's wonderful because it can measure all cell types, it measures single cells, but it is in fact a single time point analysis. So it doesn't give you information on what's happening on a single cell over the course of time. Now, why is it difficult on an SPR to measure a cell? This is an example now approximately drawn to scale. So if this is a gold surface of an SPR sensor, and you think about a cell that is about 10 micrometers in diameter, the sensing field, so the SPR field that emanates from the surface, has a decay length actually in the 100 nanometer range. So it decays very rapidly into solution and it doesn't even see the whole cell. So the morphology of the cell, whether it's flat or round, and the binding signals interfere very much. And actually SPR can't detect any binding up here, but only down here. And then there's another very crucial problem with SPR sensors, and that is that usually you want to apply a high flow rate in such a cell, in such a flow channel, in order to prevent measurement artifacts. But if you apply a high flow rate, then the cells shear off the surface. So if you immobilize them before, and if you apply a high flow rate, you will simply lose the cells. Now, what we're trying to do here is now we're trying to reconcile these two fields. The biosensors, biocores have been invented in the 1980s. I mentioned that. And there have been quite a number of attempts to measure cells on SPRs, but they have only been successful in an academic setting, I would say with a lot of hands-on work, but not in an automated setting that would also translate to the pharmaceutical industry. And the flow cytometer, of course, super automated, very successful, also a considerably larger market. But as I mentioned, no real-time measurements are possible at this point. Now we invented this cell trap technology, and I'd like to show you this simple movie of how this actually works. So again, we're using our microfluidic chips, but now you will see they are modified. They have a cell trap on them. The samples are again being administered to the instrument here in the lower sample compartment. And this is also where we place the cell suspension. And now when we zoom in to our chip, you can see the microfluidic flow channel here. And again, our two microelectrodes, 100 micrometers in diameter. And now they have been functionalized with a trap, a plastic biocompatible material. And now a cell is being aspirated from the cell solution and being flown into the trap and is physically retained in the trap. What you've seen now, just the pink molecules, are the fluorescently labeled antibodies that are being flown across the cell surface and are allowed to interact with our cells. And then again, very similar to what we've seen before, we wash off the antibodies and allow for dissociation. And then this is when we measure the dissociation of antibodies and the stability of this interaction. Because our instrument can reverse the buffer flow, we can also remove a cell and transport it to waste, and then measure the next cell. So here are some scanning electron micrograph images of these cell traps standing on our microelectrodes. You can see here we can vary the dimensions of this. We can vary the window size, and these are essentially polymer cages that are 3D printed onto our helix chips. Now, here are some more images you can see here. This is for calibration purposes. This bead here is not a cell, but it's a plastic bead, 15 microns in diameter. You can see that we can tailor the size of these cages to accommodate just a single cell. So it doesn't have to be 15 micrometers, it can be 5, 10, 15, 20. So actually we have cell traps of different sizes. And you can see when we look at the cell traps from above, these are examples of chips that we prepared with one cell trap where we only measure a single cell, or 3 or 5 cell traps where many of these cells are measured at the same time. Now, the cool thing is you can measure with fixed cells, but you can also measure with cells that are alive and living. Now, how is the measurement being done? You've already seen that in the video. At first, the cell is being injected. It's being held in the cage. What the instrument does is it takes a snapshot of the empty cell, then captures the cell, takes a snapshot of the cell in the trap, and then it injects the analyte molecules. You can see here then the increasing fluorescence signal, and then flows buffer across it, decreasing fluorescence. So we get the on rate from that phase, we get the off rate from the dissociation phase, and then we eject the cell from the waste again. Now here's an example from real measurement data that shows different cell types, CHO cells, embryonic kidney cells that are captured here, and how these are being, how this measurement signal looks like. Here are 3 consecutive injections on the same cell, and then 1 dissociation that's being performed here. And the on rates and the off rates and the KD values can actually be recorded with high confidence. Now, I think I'm coming to the end of my time, so I will just show you maybe 1 or 2 examples of how this cell trap can be used. This example is from a collaboration with Amgen. Here is they are studying bispecific T-cell engagers. So these are antibodies, engineered antibodies that would bind to a target cell, usually a tumor cell, and a T cell at the same time. And we have analyzed different BiTE formats. You can see here a BiTE that binds to the T cell here and dissociates from the T cell, and then also a BiTE that the same BiTE binds to the also to the tumor cell. This is another BiTE that only binds to the T cell but doesn't recognize the tumor cell. And we've done that for many different formats. And you can see here that the technology can be used to select the most promising candidate. So you can see here, these are BiTEs, again, for that all engage with the T cell in a very similar manner, all single-digit nanomolar KD values. And then this is a variation of different binding moieties with respect to the tumor cell. And you can see these 2 are actually the same, giving very similar KD values. But this one here would be the one that would be advanced in the drug discovery pipeline because it has a retention time on the tumor cell that is 4 times longer than the other binding moieties. And this is now measured on a cell. I think I will come to an end very, very soon, just to give you an example about you can also discriminate binding modes. So this would be the discrimination of an affinity binding where an antibody engages only via one arm, or whether an antibody engages via two arms. So you can then analyze rate constants, dissociation rates, or binding stabilities from these one-arm engagement versus the two-arm engagement. And I think in the interest of time, I will maybe skip the last part. I would just like to mention here the technology has been used, the kinetics have been used to identify whether a GPCR on a cell would actually cluster on a surface. So antibodies have been used in a bivalent format and as Fab fragments as a monovalent format on GPCRs that were expressed at high densities and low densities. And we could actually show in this case that even at low densities, the GPCR would cluster on the cell surface. It would form clusters, and that was actually a very important structural information that could be obtained from the binding kinetics here. So it's not just about binding kinetics. Sometimes the kinetics can actually tell us something about structural properties on these cell surfaces. And with that, I'd like to close. Thank you very much for your attention. I hope it was not boring for you. And I hope I could show you a couple of examples where SwitchSense and CellTrap provide interesting information. I could not go into details of enzymatic activity and conformational changes for time reasons this time, but we'd be very happy to discuss these or other applications in the future with you. You can always contact us at info@Dynamic Biosensors or talk to Takaguchi-san, who represents us in Japan, or you can contact me directly. So thank you very much for your attention.
Thank you very much for interesting and very exciting. So, firstly, I would like to gather some comments from the audiences. So, 皆さんコメント、質問どうでしょう？いかがでしょうか？挙手していただけるとありがたいんですけど。はい。では、最初私から。じゃあどうぞ。井本先生どうぞ。最初。
Thank you very much for your presentation. It was very impressive, especially at the end of the talk on CellTrap system. But so the CellTrap itself is really exciting. But how about the kind of information on the volume or sample consumption, or how much cells you have to prepare, and also the regarding to other previous conventional technology, how much sample you require to do the experiment, and how much throughput you can achieve at the highest throughput. That kind of information will be very appreciated if you could.
Thank you very much for the question. So I will answer the last question first. So regarding the throughput, you've seen that one of these machines has a 96 or 384 well plate format. This is how the samples are being given to the instrument, and it can measure one chip at the time, but it has an automatic chip exchanger for up to 5 chips. So typically, you would have a throughput of 1 to 2 plates per day with such a machine. But the idea of these machines is that they are being combined, not just used as one module, but they all have an embedded PC and they are connected to the network of an institution. And so anyone in the office can then, via the network, access these machines and send workflows to different biosensor modules, different Helix modules that are in the network of this institution. So we, for instance, our own scientists here, we have always 5 to 10 machines in our application labs, and they are rarely seen in the application labs, our own scientists, because they mostly communicate with the instrument via our network, and then they use not only one machine, but also several machines at the same time to increase the throughput. So with a typical, for instance, a 4-helix bundle that would, in terms of price, would be similar to a Biocore 8K, for instance, instrument. So that's in the $400,000, $500,000 price range. You could reach a throughput of thousands of samples per day, more than 1,000, depending on the dissociation times that you're using. So we've seen also in this presentation, you've seen some molecules dissociate within a very short time, within a couple of seconds. So the instrument can measure many molecules sequentially in a short time, whereas other molecules like antibodies that are already tight binders, they take many minutes or hours to dissociate, and then the throughput goes down essentially. But the idea is really to create a cloud of biosensors with these helix modules and combine many of them in a very versatile way. In terms of sample consumption, we're using actually very, very low amounts of samples. So you've seen that typically we have a sample preparation step before the measurement. And during this preparation step, a protein is conjugated to a DNA strand. And then it depends on the efficiency of this reaction, but typically we obtain a conjugate that is good for hundreds of injections on the sensor surface. So sometimes we do a conjugation reaction and you can run 700 experiments. Sometimes a conjugation reaction will only yield 50 experiments. But the idea is always that you take these conjugates and you store away what you don't need in a freezer, and then a couple of days later or weeks or months later, you can then thaw from the same batch again your DNA protein conjugates and use them on the sensor without any new sample prep step. And in terms of sample consumption, these are really very, very low volumes. So typically we use a couple of microliters, and the concentrations are in the typically 100 to 200 nanomolar range for such an injection. So these are small and low sample volumes.
Thank you very much.
Any other questions or comments? So then I have many questions. Okay. Yes. So first of all, I would like to ask about adapter design. Very specific talks was very important. You have talked about the design of an adapter in the first part of your presentation. So someone might be wondering how we choose the adapter and how we design adapter. Please give some advices on these topics.
Yes, thank you very much. I will bring up one of these slides again. Yeah, so this is essentially what Tsumoto-sensei has been referring to. So this is the adapter strand and the anchor strand and the ligand strand. So in our standard configuration, this is 48 base pairs, is a mixed sequence. And these sequences here are also non-confidential sequences. So we share them with customers, and typically you would use a 96 base adapter strand and a 48 base ligand strand. But this could also be a shorter or a longer strand, depending on what you would prefer in your experiments. So for most experiments, a 96 base pair is a good compromise, because it's about 30 nanometers in length, and that keeps the molecules away from the surface and makes them accessible well. At the same time, these interactions here, so if you have a 48 base pair DNA duplex, that is very stable. So you don't have to worry that this would come off during an experiment. It has practically an infinite off rate on our timescales that we observe here. And sometimes we have, I couldn't go into this in more detail in this talk, but when people do not use a protein or small molecule ligands, but actually use RNA sequences or DNA sequences that extend from here, then they are sometimes concerned about the sequences down here. So what the instrument always does is it records electrode 1 and electrode 2 at the same time, and you can then see if you get unspecific binding to the anchor strand or to the adapter strand. And if you don't like that, then you would be able to change that sequence if that's required. Yes.
Okay. My second question is about the threshold of these detection systems. Could you tell me the limitation of the lower K-off and higher K-off and high K-on and?
Oh yes, absolutely. Yeah. So I've just mentioned the off rate here. I've shown that with respect to a fast K-off. Yeah, so that would be one limitation, and you can see here that we could not measure the 3 base pairs perfectly anymore. I would say we could measure with confidence the 4 base pair, and that is about 20 per second in dissociation rate. So that would actually be a very, very fast dissociation rate. And the reason why we can measure that is, first of all, we use a 100 Hertz sampling rate. And I did not really explain that in detail, but if you look at the biochip, so the fluidics of these biochip are quite unique in that you can run a sample from one direction across the biochip, and then there's a couple of valves directly under the biochip that will switch the flow direction from left to right to left. So you can reverse the flow direction very quickly. And by this, we can get washout times of 20 milliseconds. And that is really, really fast. This is why we can measure these fast off rates in case they are needed. On the other side of the spectrum, on the slowest off rates that we have measured so far, I think pertain to the streptavidin biotin system with femtomolar KD values. And the off rates that we've seen here were 10 to the -5 and 10 to the -6 per second. So very slow dissociations measured typically overnight over 8 to 12 hours, but that is also possible. Yeah.
Yeah, maybe this point would be very advantageous than the biocore system, because the biocore system has limitation on the K-on value and K-off value. So your system complements these non-measurable area in biocore world. So this point is one of the most important topics for general biochemist. Yeah. So the next question is about the biospecificity. Yeah, I'm very surprised to see today's results, showing the complete perfect biospecificity of one antibody, Hemvida from the Chugai. So I want to know another case on amivantamab. This biospecificity antibody also very promising, because this is well organized and well designed. So could you tell me the result of the interaction analysis of this amivantamab? Is available?
Unfortunately,
from Janssen.
Yes. Yes, you're referring to the upper one of these antibodies. Unfortunately, we have not measured this antibody in our lab yet. We have the case study that I showed yet was on Hemlibra. We have measured quite a high number of other bispecific antibodies and also trispecific and tetraspecific antibodies together with pharmaceutical and biotechnology customers, but unfortunately we cannot share the data. We can only share the data on available antibodies. So we have also measured for other antibodies. We have measured bispecific BiTE and antibodies that are also quite interestingly very much like Hemlibra. The BiTEs are rather on the weak side, whereas the amivantamab is a stronger antibody for sure. But unfortunately, I don't have data on amivantamab as of yet.
Yeah, recently a lot of antibody engineers have recognized the importance of this bispecificity by using biophysical analysis, because this analysis would strengthen the evidence for cell actions. But you know, some failure has already been reported in the bispecific antibodies, showing that this bispecificity cannot be shown on cells. So yeah, this data will strengthen the power of the bispecific antibodies for development. So maybe a lot of the researchers in pharmaceutical companies have great interest in this system, right?
Yes, I couldn't agree more with your comment. I think, first of all, the binding modes of these antibodies are highly interesting and are also highly controversial. I agree with you. Even when we measure this now on a biosensor surface, it doesn't mean that on the cell surface it will have the exact same binding modes, because it will depend on, for instance, the accessibility and the fluidity of and mobility of the different targets on the cell surface. All these things are important. So I agree with you. I think it's a huge potential, but the binding modes are quite poorly understood so far. And what's also interesting for me when we looked into the development of Hemlibra, which was, I think, a wonderful work by Chugai, really very impressive. But it's interesting that they actually brought the bispecific antibody to the clinic and to the market without even knowing the exact specifics of the binding modes back in the day when they had to do it. So I think the enabling tools that were available were quite limited at that time, and still they achieved this wonderful result. So very impressive.
Yeah, I see. Thank you very much. Any other questions or comments? Oh, I have more questions. So, yeah, next question is about GPCR clustering. So the biophysicists have recognized the importance of this clustering effects. So, but no one can experience such a great result as this. So how we can emphasize on this importance? So
yes. Yes, so in this case, we were lucky again to partner up with a big pharma company that remains unnamed, but they allowed us to share some of these results here. The way that the clustering was being investigated here is, and sorry for being so fast in that respect, is they had already prepared antibodies in a bivalent form and in a monovalent form. And what we did then is we compared, you can look here at the dissociation rates of these antibodies. So we had immobilized the cells here that had been pre-incubated with these, either the full antibodies or the Fab fragments. And you can see that the Fab fragments, these in blue here, they dissociate rather quickly from the surface. Whereas the bivalent antibodies, they have this very slow dissociation that is a strong binding mode. And that is, by the way, I've shown this for the example of an antibody that we ourselves bought, an anti-CD3 antibody, where we could identify the binding via one arm and two arms. So we can, and we see that, by the way, we see that very often in many antibody formats, that we see a weak binding mode and a strong binding mode, and we can then ascribe the strong binding mode to the engagement via two arms. And that is consistent with what we see here. Now, the interesting result here was that different cell lines were being tested. And one cell line had a very high expression level of the GPCR, whereas the other one had a low expression level. And what we would have expected that at the low expression level, so for the Fab, both curves should always look the same. It shouldn't matter on the expression level. But for the bivalent analyte, for the full antibody, we would have expected that the low expression would be similar to the blue curves here, but it did not become similar, and we have more extensive data sets on that. So from that, we could see that even when the GPCR is expressed at very low levels, almost beyond the detection limit, especially of flow cytometers, so it must have been very, very low, we could see that the antibody engages with both arms. And that means that they're either clustering or they're very mobile, and they move into the vicinity so that the antibody can grab onto two GPCRs at the same time. And that was kind of, I think one mustn't overestimate these results. I think it's one element in understanding the whole system, but I think it's definitely a very interesting finding here, yes.
Yeah, so several researchers are wondering whether clustering can be measurable or not. But your system clearly shows that at least clustering can occur. So then this would be very important because clustering might strengthen the function of these receptors. So then, but no one measure directly the interaction of the clusters receptors. So your system clearly shows that the clustering dominates the interaction on cells. So this is very, very, very exciting for me.
Yeah, thank you. We're also very excited, and actually just to maybe add to that, we are looking. So the cell trap technology has not been launched by us yet. It will come to market by the end of this year in December. But we are very open to collaborations in the meantime and to do beta testing with that. And we would be very happy to, have more people trying the cell trap in the meantime.
Yeah, I have been investigating protein interaction many years, so I know very well about the failure of the detection on cells by using SPR. You have shown that the biocore system cannot be applied to this detection. So I have experienced this. So this result is very, very encouraging for us because cell surface interactions can be applicable. So by using your systems. So yeah, I will be try. But could you tell me that this system can be applicable to the former machine style, right? So
yes. So these
I have one machine, but I want to apply your system.
Okay. Absolutely. So what we need to do for the Helix is there is a special calibration required. And then there's a special software package that needs to be installed. But this is, in principle, the Helix machines are going to be compatible for these cell measurements. So we'd be happy to try this with you in your lab. And yes, we should arrange for that. I would be very, very happy and grateful for that. Thank you.
Yeah, thank you very much. So any other questions or comment? 何か質問かコメントありますか？ぜひ、リアクション。ないですね。So finally, I would like to know about how many machines have already been introduced in pharmaceutical. Yes.
Yeah, in total, we currently have more than 60 machines in the market. but part of that are the first generation biosensors, and part of that are the second generation, the Helix systems that you've seen here now. But we have seen, fortunately, I'm very happy to say that we've seen very good adoption of the Helix system. So we're currently we will be moving into bigger office and production spaces next month in Munich, because we are currently increasing the production of these instruments and also preparing for the cell trap launch. So we are currently very happy that we can support our customers well.
Oh, thank you very much. ありがとうございます。Any other questions or comment? よろしいですか？今先生、よろしいでしょうか？
ありがとうございます。はい、ありがとうございました。大丈夫だと思います。
Okay, so then we would like to close this session. Thank you again for your very exciting and nice talk. Thank you very much.
Thank you. Thank you very much for your attention, and thank you for the kind questions. Thank you.
はい、では、湯本先生、最後お願いします。
どうもありがとうございます。では、すいません、私、今回ではないんですけれども、全体の、この勉強会の方の、企画に関わっております、湯本です。今日は、ラントさんにご講演いただきましたけれども、来月もですね、月末の金曜日になりますけれども、ご講演を依頼しておりまして、遺伝子解析の最先端の情報になろうかと思うんですけれども、そういったものを今後も引き続きですね、企画しまして、このコミュニティの皆様にご案内していければというふうに思っております。今日は津本先生、どうもオーガナイザーありがとうございました。また、Thank you very much for your great presentation. We really appreciate your input to the community.
Thank you very much, and thank you for taking the time. I appreciate it very much, and have a nice weekend, everyone.
Yes, we do.
Thank you. Bye bye.
GmbH の矢田様、すみません、最後お願いいたします。
はい、ありがとうございました。本当に最後まで皆さん、じっくりと聞いていただけたと思います。今日金曜日ですけれども、ゆっくりと今日のお話をかみしめてお過ごしいただけたらと思います。木本先生どうもありがとうございました。木本さんどうもありがとうございました。先生どうもありがとうございました。Thank you very much。皆様、それでは、良い週末をこれにて失礼いたします。ありがとうございました。失礼いたします。
ありがとうございました。
Auf Wiedersehen. See you later. Dankeschön.
Dankeschön. Thank you very much.
Thank you very much.